Hello everyone, and welcome to episode 123. In today's show, we'll be continuing our mini-series on the Malacostracans, a biodiverse and ecologically irreplaceable class that contains the vast majority of all living crustaceans. In the first part of this mini-series, we explored the mini-clades of parasites, bottom feeders, and shrimp-like creatures that populate the Malacostracan ranks. In fact, we looked at virtually all the Malacostracan orders, with one major exception: the decapods. The reason I set the decapods aside for later was because this is a massive and wildly successful group with many unique descendants, and they required not just one but 2 episodes of their own for further analysis. So today, we're going to dig into the decapods by exploring their basal clades, their intermediate clades, and one of the major derived branches, the Astacidea, also known as the true lobsters. And in the next episode, we'll look at the most derived group of decapods, the Brachyura, or the true crabs. These are really fascinating creatures, so this and the next episode should be just as exciting, riveting, and revolting as the previous episode. Okay, so with that said, let's get started. Our story begins at the dawn of the Devonian period, about 420 million years ago. By this time, the Earth's oceans were filled with all manner of creatures, protostome and deuterostome alike. There were primitive fishes, from the early worm-like jawless fish to the more experimental permutations, like the Acanthodiae spiny sharks and the placoderm armored fish, and the earliest Actinopterygiae ray-finned fish. There were also the aquatic arthropods, like Eurypterids, Trigonotarbids, and ancestral myriapods creeping along the seafloor. Mollusks, including primitive snails and cephalopods, were also proliferating. And across the entire water column, from the sunlit shallows to the open ocean to the darkest depths and to the ocean floor itself, there were crustaceans. Countless brachiopods and copepods swam in the sea and crawled on the sand, while all manner of primitive barnacles colonized reefs, rocky shorelines, and intertidal zones. The Devonian marine ecosystems were rich and biodiverse, flourishing with life that had originated, diversified, and spread out across the preceding Ordovician and Silurian periods. These earlier crustaceans played a foundational ecological role, as many of them, like the copepods, were just tiny zooplankton that saturated the water, or they were a bit larger but still numerous, like the krill, and these provided an organic substrate that could be filtered out and fed upon by an entire ecosystem of larger organisms. If we roll back the clock a little bit and go to about 460 million years ago in the Ordovician, the earliest divergence from the rest of the crustaceans had begun. This new offshoot was small and not very biodiverse. A couple basal sublineages spawned quickly before the close of the Ordovician about 443.8 million years ago, but after that, the rest of the decapod stem group stabilized but didn't really proliferate and diversify throughout the Silurian. But things began to change around 420 to 400 million years ago at the dawn of the Devonian. At this point, the stem group decapods had not just emerged, but were beginning to diversify and radiate. As this crustacean offshoot came into its own and all these new families emerged, they carried with them a basic decapod body plan, the inherited caracoid facies, or the shrimp-like body form. Of course, this was altered slightly in some groups, there's always some variation, but for the most part, the decapod body plan was, and is, fairly consistent. This body plan includes 3 major body segments: the head, the thorax, or the pirion, and the abdomen, also called the pleon. In this caracoid facies body plan that's so common among the decapods, the head is equipped with a number of forward-facing appendages, including pairs of antennules and antennae for sensing the world, sometimes eyes mounted on short stalks, although many species have evolved to lose their eyes. And mouth parts, such as mandibles and 2 pairs of maxillae for handling food. The head and the thorax are typically fused or integrated in some way, forming a larger body section with 10 segments. They're equipped with a carapace, an extra armor plate on the exoskeleton that straddles the head and the thorax like a cloak. A portion of the carapace, called the rostrum, extends between and beyond the eyes, like a crest or a nosepiece on the helmet of an Iron Age warrior. Numerous limbs protrude from the underside of the thorax and the abdomen. The first 3 pairs of limbs are the maxillipeds, which are legs that were evolutionarily shaped into extended mouth parts. They assist in feeding and handling food, like holding down prey as it's chewed on, or in the case of species that filter out their food that filter feed, the arms may have comb-like components that can be used to filter through the water and catch smaller particles that can then be scraped off on the mouth or fed to some of the maxillae. The next 5 pairs of legs are mounted under the thorax. These are typically long and slender and extend forward and down, but in many species, like crabs, the legs have evolved to be bulky and spread out laterally. These 5 pairs of limbs are really important for the decapods. Not only are they used for locomotion, they're also often equipped with 1 or more sets of claws to assist in crawling around, catching prey, fending off predators, and interacting with mates. More importantly, many of these pairs of limbs also bear sexual organs and gills. Frilly or layered gill structures exist near the base of the limbs, almost like armpit hair. And for those species that sexually reproduce, on the females, the 3rd pair of legs are modified into sex organs. And in males, it's either the 5th pair of legs that's been modified into sex organs, or it's the nearby 1st pair of pleopods. The pleopods are swimming legs mounted on the abdomen, or the pleon. These pleopods are like stumpy little legs with small fins, and they're used to help propel the decapod through the water. In some species, they're used to brood eggs, and they may even be equipped with additional gills. Similar to the cephalothorax, the abdomen also has 10 body segments. The abdomen is tipped with a telson, or a small tail. Depending on the species, the telson may be a small point, a number of narrow spines or barbs, or it might have a laterally flattened appendage that forms a tail fan, similar to the tails of cetaceans, like whales or dolphins. One major evolutionary adaptation to this body form, which I'll get to later on, is seen in the true crabs and other crab-like animals, where the abdomen has been deflated almost and tucked under the thorax. Alright, so this is the general decapod body form, this caracoid facies, this shrimp-like body shape, which you should keep in mind as we go through the episode. With that said, let's return to their evolutionary history, when the decapods really began to diversify and proliferate heavily in the Devonian period, about 420 to 400 million years ago. Like many of their contemporary cousins, the individual organisms making up these fresh, new, divergent lineages were also relatively small, akin to krill and other shrimp. Some of the more basal clades of the decapods are similarly diminutive, and also similar in morphology and ecological role. For example, the most basal decapod lineage is the Dendrobranchiata, also known as the prawns. Another basal lineage would split into the true shrimp of the Caridea and the boxer shrimp of the Stenopodidea. These were the 2 offshoots that branched off quite early, after the decapod lineage had established itself, but before the Ordovician had really come to an end, and they tell a really interesting evolutionary story. The Caridea, or Caridea, the true shrimp, are roughly similar in morphology to the prawns of Dendrobranchiata. The overall body form is angular and sleek, with a narrow, conical head equipped with long antennae, 2 eyes mounted on short stalks, and a humpbacked thorax midsection with a cluster of long, narrow legs angled forward and down. These body sections are fused in an integrated cephalothorax structure with a protective carapace on top. And there's a smoothly tapering abdomen that flares out at the end with the crustacean version of a tail fin. And both the true shrimp and the prawns have species that grow anywhere from just a few millimeters to well over a foot in length. Ecologically and behaviorally, there's also tremendous variety within both the Caridia and Dendrobranchiata. They both have member species that live in the shallow continental shelf area, in reefs, in tide pools, and those species that live in deeper waters, in ravines and trenches, and those that burrow into the seafloor itself for shelter and food. Both groups are predominantly omnivorous, eating anything suitable that they can find, although there's generally a higher proportion of filter feeders and algae grazers among the Caridia. And both groups, of course, have high biomass populations and represent an irreplaceable food source for larger animals like seals, penguins, seabirds, and whales. But if you start to look closer, you might begin to notice subtle differences between the prawns and the true shrimp. One of the big subtle differences between the true shrimp and the prawns is their gill physiology. The more primitive Dendrobranchiata are named after their gill structure, which is fluffy and tufted and branching, like the canopy of a tree. In contrast, the true shrimp have lamellar gills, where the tissue is arranged in sheets or rows. This more developed gill pattern increases the surface area available for gas exchange. Another major functional difference can be seen in their legs. The prawns have 3 pairs of legs equipped with slender claws for grasping around and handling food. The true shrimp have lost a pair over time, and they only have 2 pairs of claws. There's also a not-so-subtle, visually obvious anatomical feature that you can use to tell them apart. The prawns have a more linear body shape, whereas the true shrimp have a bulging second segment in their abdomen that bends their tail downward. This morphological feature is called the Caridian bend, and it's actually a really useful diagnostic used by fishermen and scientists alike. On that note, the overall body form of the shrimp, or a collection of general traits that make up this classic shrimp body form that we've seen throughout all of the crustacean clade so far, is called the characoid facies. It includes stalked eyes, branching or biramous first pair of antennae, an abdomen that curls downward to some degree, and a caudal fan, the crustacean equivalent of a tail fin. Perhaps the most significant difference can be seen in their reproductive cycle. The primitive Dendrobranchiata perform brief courtship rituals, and then, after mating, the fertilized eggs are released during the night into the water column, and they just float away, easy food for other animals. But lots of eggs are produced, and by playing the numbers game, it's quite likely that at least some eggs will survive to hatch into live offspring. In contrast, the true shrimp retain their eggs and brood them. When the prawn eggs hatch, the newborn is a nauplius larva, which is a life stage unique to crustaceans. This young nauplius larva is composed of just 2 or 3 body segments. There's a head and a tail. Maybe in some species there's a middling thorax or a carapace plate. This head-tail body form is covered in hairs, and it has a single cyclopean eye. This naupliar eye is typically lost during later stages of development, as the body radically transforms. The larva will undergo several molting cycles, each time adding body segments and producing more developed limb structures. When it can start swimming with its new thoracic legs, it enters the zoea stage, and after that it becomes a juvenile with the mature body form. Now, in the true shrimp, the larva goes through these naupliar stages in the egg, and it hatches in that later zoea stage. As a result, the offspring mortality rate of the true shrimp is much lower, because the eggs are protected and brooded instead of dispersed immediately into the water column, and the larvae that hatch are larger, stronger, and better able to move around to find food and avoid predators. Do you remember that I mentioned another clade earlier? It was related to the Caridia. These were the boxer shrimp of the clade Stenopodidea. Like their true shrimp cousins, these boxer shrimp also have lamellar gills, and they also brood their eggs. However, they've retained 3 pairs of claws, like the more basal Dendrobranchiata, and something really interesting happened to that 3rd pair. In the Stenopodidea, the 3rd pair of walking legs and the claws at the end have been enlarged. They're disproportionately gigantic, and armored, and spiky compared to the other legs, and the claws at the end are thick and powerful, much heavier and more laden with muscles than the other smaller pairs of claws on the more slender legs. Consider the banded cleaner shrimp, Stenopus hispidus, which lives in reef environments in temperate to tropical waters. In these habitats that are usually characterized by a rich community, they'll crawl out onto a patch of coral or some rocky surface and expose themselves by lazily waving around their antennae. Their antennae are like long white whiskers, contrasting brightly against the dark rock and the gloomy water. By waving them around, the banded cleaner shrimp is advertising itself, showing off to other fish in the community who may be interested in its cleaning services. With its 3 pairs of claws, including the enlarged pair mounted on the 3rd pair of legs, the banded cleaner shrimp will pick off any debris or contaminants, such as ectoparasites like isopods and brachyura, pieces of fungus, or flakes of dead epidermis like loose scales. This is how the cleaner shrimp gets food. It eats all of these parasites and this fungus and all of this debris and stuff. It eats it like snacks. It just gobbles it up. And because it provides such an important service to other individuals in the reef community, they basically endear themselves to the other species living around them, and in this indirect way, they can avoid predation. Even carnivorous fish will come up to the cleaner shrimp, and instead of eating them, which would be easy, they too will expose their teeth to let the cleaner shrimp get out the gore and leftover flesh from their kills, and they'll let the cleaner shrimp pick out parasites from their gills and their scales. This ecological service strategy is seen in many other Stenopodidean species, like the ghost boxing shrimp Stenopus pyrosonatus, the relatively shy gold coral-banded shrimp Stenopus scutillatus, the Pacific cleaner shrimp Lysmata amboinensis, and the Eurocaridella antenbrunii, which is known to feed at night by cleaning the teeth of sleeping fish. Now that really seems like a risky strategy, but they've evolved to do it quite well. Even predators like sharks and eels will let them come close while they sleep. Now, all of these groups that we've discussed so far, prawns, the boxer shrimp, the true shrimp, they're all grouped together informally as the swimming decapods. When these very basal decapods diverged about 460 million years ago and diversified over the next 40 to 60 million years, the remaining decapod lineage they left behind would go on to form the Reptantia, or the crawling or walking decapods. Of course, despite their names, their means of locomotion demonstrate a ton of variety in both groups. Mini boxer shrimp, for example, are in the swimming decapods group, but they crawl around. Most of the so-called swimming decapods actually spend a lot of time on the ground, in their burrows, digging holes, or hiding from predators, or trying to maneuver around to acquire or handle prey. Similarly, there are a few Reptantian walking decapods that swim, sometimes in short bursts, sometimes more. This naming convention of swimming and walking decapods was technical terminology in the earlier half of the 20th century, but it was formally discarded in the 60s, and now it's little more than a loose generality that has lots of exceptions on both sides. Anyways, this Reptantia lineage would find itself well suited to its marine environment. populations would grow and expand, radiate into new marine habitats, like new trench systems, or new expanses of sandy ocean floor, or in areas closer to the continental shelves that could explore and colonize new coral reefs, new kelp forests or seagrass meadows, and intertidal zones. Around 390 to 400 million years ago, so still in the earlier part of the Devonian, a major branching event created two main lineages, whose descendants would be immensely successful and highly diverse. In the modern day, the Reptantia includes about 11,000 species across both lineages, although around 7,000 of them are brachyuran crabs. All told, Reptantia makes up about 3/4 of all decapod species, which itself makes up a little under 1/4 of all crustacean species. Let's focus on one of these two main lineages within Reptantia, which emerged so long ago in the heart of the Devonian. Sometime in the late Devonian or the early Carboniferous, in the time window of about 340 to 370 million years ago, a branching subgroup called Acheilada evolved to live in the deeper ocean, away from the intertidal zone. They burrowed in the sediment and evolved to lose their claws, which is the strange and unique feature for which they're named. Acheilada means without claws. Along the way, the achelates also evolved a unique larval stage called the phylosoma, wherein the larva is flat and transparent. Like so many other crustaceans, the larval form is free-swimming, and as it's tiny, it's part of the zooplankton community. The phylosoma is unique because it's relatively unusual looking for a crustacean larva, with its strange, flat body, its eyes on long stalks, and its very long, slender legs, all coolly transparent. They're large, too, standing out among their fellow zooplankton as big, gangly weirdos. Metamorphosis out of the phylosoma stage represents a radical transformation, as the entire body is reshaped into the adult form, with a larger abdomen, reconfiguration of the legs, adjustment of the eyes, and a reshaping of the entire exoskeleton and carapace. The adult Acheilada have the same general caracoid facies, shrimp-like body form, except they're bigger, like way bigger than any shrimp we've talked about so far today. They look like lobsters, except for their strange antennae, and of course, the conspicuous lack of claws. Depending on the species, adults can grow up to 40, 50, even 60 centimeters long. They have hard, thick carapace armor and robust appendages. Their legs are arranged not so much underneath them, but more so like a spider or a scorpion's legs than a shrimp. They're angled outward to the side, they're more laterally oriented, as opposed to being clustered directly underneath the body. The Acheilada live on the seafloor, where they scavenge for detritus and smaller animals that make for easy meals. Within the Acheilada, there's two groups, the spiny lobsters and the slipper lobsters. Spiny lobsters live in crevices, reefs, coves, trenches, and other protected, sheltered areas along the seafloor. They generally prefer warmer areas, like tropical and subtropical waters, where they live in large social groups. These groups have really interesting social dynamics that offer fascinating hints about the social lives of the spiny lobsters. For example, they like to stay near each other, in close contact, and they use their very long antennae to feel around for each other, to know that their friends are close by. Sometimes they'll migrate as a group to a new area, forming a long single-file line following a leader. During the day, they're mostly hiding in small crevices and sheltered spaces, and during the night, they'll come out to hunt smaller, softer, and slower prey, like urchins and bivalves, and aspid sea hares and sea snails. Captured prey is awkwardly, remember they don't have claws, it's awkwardly carried back to shelter to be eaten in safety. The other clade in Acheilada is the slipper lobsters. These are bottom dwellers that live about half a kilometer deep, where they feed on scavenged bivalves, crabs, starfish, bristleworms, and sea slugs. The slipper lobsters are relatively young, with the lineage emerging a mere 130 to 150 million years ago, although genetic evidence suggests that it may be as much as 200 million years old. These slipper lobsters have adapted to live in the mud and sediment along the bottom of the seafloor. Their tails are not long, but instead are stout and tucked, making them look from above like stubby thumbs or oval-shaped dinner plates. Their armored carapace is thick and broad, descending along the edges to produce a thorny frill, protective edging that can help hold sediment on top and secure the slipper lobster when it burrows into the muck to hide. The most visually obvious and taxonomically distinctive feature of the slipper lobsters is their antennae. The spiny lobsters have long, thin antennae, which they use in part for social communication. The slipper lobsters, being mud-dwelling weirdos, are less social, and instead, their antennae have been adapted into short, broad, flat, disc or fan-like extensions. The antennae are like strange Mickey Mouse ears, or pedals, or like ping-pong paddles. They're good for wiggling and sifting through the soft mud in search of food, or for lifting up the mud to bury themselves within. In effect, the slipper lobsters have adapted their antennae to be a little more physical, a little more helpful when it comes to digging and mining and burrowing through the mud. Shortly after the Achilata emerged, the remaining stem group underwent another branching event to produce the Polychelida and the true lobsters of the Astacidea. Now, the Polychelida is a small group with fewer than 40 known species, and all of them are deep-water, benthic organisms that hang around on or near the seafloor. Like other members of this branch of Reptantia, their ancestors lived in shallower water with vegetation and high community densities. But eventually, these groups descended to lower waters, up to 5,000 meters, and they adapted to burrowing or sheltering at these great depths. With respect to the polychelids, even their eyes have shriveled away to nothing, as they spend most of their time in the darkness of the mud and the shadows of the rocky seafloor. Instead of eyes, these blind crustaceans just have empty eye stalks, or no eye stalks at all. Where the Achilata are named after their lack of claws, the Polychelida are named after their abundance of claws. The first 4 pairs of legs are equipped with claws, and in some species, all 5 pairs of legs are equipped with claws. Consider the Polycheles typhlops, a species of polychelid that can be found in benthic and deep water communities across the world's oceans. They hide from predators by burrowing into the sediment and raising their enlarged front pair of arms to guard the sides of their head. The posture also keeps their most powerful pair of claws up and at the ready, hidden by a thin layer of sediment so they can quickly react and strike back against a surprised attacking predator. Okay, so we've looked at the lifestyles and ecologies of many different decapods, including the swimming decapods and one of the 2 major branches of Reptantia, the crawling decapods. On this branch of the Reptantia family tree, there's one more group, one more clade left for us to explore. This group is the Astacidea, which includes the true lobsters, as well as their crayfish relatives. In the next episode, we'll get into the other branch of Reptantia, which includes all the crabs, but for now, let's just take a closer look at the lobsters and the crayfish of Astacidea. This is a group that emerged in the earlier days of the Carboniferous, about 335 to 350 million years ago. The common ancestor was a benthic marine predator, a burrowing, ground-nesting crustacean that hid among the rocks and crevices on the ocean floor. The true lobsters of the family Nephropidae descend from the ancestral Astacids, and today they can be found in all the world's oceans, populating marine seafloor communities across every continental shelf on the planet. By 330 million years ago, the 2 main lineages in Astacidea had been established. you had the clawed lobsters and the crayfish. The lineage of clawed lobsters would soon split into the true lobsters of the family Nephropodidae and the reef lobsters of the family Enoplometopidae, which are smaller and generally prefer deeper water reef habitats. All told, these are relatively small clades, with less than 50 species between them. The sister lineage, that of the crayfish, evolved from a population of ancestral lobsters that adapted to freshwater habitats. They colonized rivers where they met the sea, and they spread further upstream, going deeper inland to colonize the lakes and ponds that feed the rivers. This crayfish lineage was much more prolific than the clawed lobsters. Over the last 330 million years, crayfish have diverged into 4 families, with more than 600 species. Both lobsters and crayfish prefer to live in shaded, protected parts of their habitat, either under rocks or shelves of coral, or burrowed in the sediment or the mud, or hiding under vegetation and decaying mats of aquatic plants. They seek mates with their sophisticated sense of smell and their antennae, and after copulation, the females usually carry the eggs in a clutch mounted under the abdomen, where they're secured and protected by the pleopods, the swimming feet. The astacids are generally nocturnal, and they're all omnivorous scavengers, with a varied diet that includes rotting seaweed, bacterial colony slime, and smaller animals like various mollusks, shrimp and tiny crabs, and even small fish. However, crayfish generally tend to prioritize scavenging plant detritus and preying on smaller, weaker, softer animals like worms, whereas lobsters have a diet richer in protein from a wider diversity of prey animals, vertebrates and invertebrates alike. Astacids also typically eat their own old exoskeleton after every molting event, as a way to recover at least some of the mineral nutrients and chemical energy. Speaking of molting, this is very relevant to their reproductive cycle. The female releases pheromones into the water, which might attract nearby males. If multiple males show up, they'll fight each other with their giant claws, attempting to crush or maim the other, or at least scare them off so they get the chance to copulate with the female. Anyways, the pheromones that the female releases signals to the males that she's sexually receptive and ready to molt. Her molting will commence, and the female will then approach the male with a fresh exoskeleton. The critical detail here is that the female's fresh exoskeleton is relatively soft and pliable. The male will turn the female on her back, and then pierce her abdomen with his reproductive organs. These are usually the first pair of pleopods that have been adapted into some kind of stabbing penis-like structure. But, like with most things, there's a lot of variation. If the female's exoskeleton has time to harden, then this penetration won't be possible. That's why the female molting is so important. And on the male side, if the male is fast enough and he can penetrate the female's soft exoskeleton, then his sex organ will deposit sperm into the female's pleoid sperm receptacles. The female can store this sperm for many months, over a year in fact, and she'll use it to fertilize thousands of eggs when she's in a place with suitable conditions. So essentially, she hangs onto the sperm and then goes about her life. She'll look for an area that's safe and comfortable, relatively stable. There's not too many predators. There's a relatively decent food supply around, a lot of algae, a lot of seaweed, something like that. And then she'll intentionally fertilize the eggs and develop them internally for a bit and then lay them when they're ready. Now, when the eggs are laid, they aren't just freely scattered into the water. They're immediately gathered up and clumped together with a sticky glue-like secretion, and then they're mounted under the female's abdomen. The female's swimming feet, her pleopods, will curl around the egg clutch to protect and secure them. And depending on the species and the age of the individual female, the clutch may contain anywhere from 5,000 to 100,000 eggs. The embryos will develop over the next 10 to 11 months, until they eventually hatch into cryptic larvae. The larva will molt dozens of times in the first couple years of life, existing in a kind of planktonic state at the surface of the water. At first, the larvae don't really resemble lobsters at all. They're what biologists call cryptic. It takes several molts before the Astacid morphology becomes recognizable. This larval period in early life is extremely dangerous. Less than 1% of the newborn hatchlings will survive this stage to get big enough where they can descend to the seafloor or the riverbed and mature into full adults. This maturation process can take close to a decade, and until the Astacid is big enough and strong enough to fend for itself, it will live very quietly, obsessively hiding in shadows, behind rocks, and under the sand. Easily the most visually characteristic feature of the Astacids is their claws. They have an enormous pair of claws on their foremost legs. The exoskeletal casing around the claws is often voluminous, making room for all of the thick bands of muscle inside that produce the powerful grip or pinch force. Lesser known of the Astacids are the smaller claws they have on their second and third pair of legs. These secondary and tertiary claws are mostly used for locomotion. The closed claw can be walked on like any other foot, but it can also pinch under rocks or marine plants and provide a bit more traction or stability. These smaller claws can also be recruited in moments of desperation as miniature weapons to fend off predators or to hold down thrashing, wriggling prey. The lobsters use their claws to attack small fish, small crustaceans, and small mollusks, including shelled mollusks like snails and bivalves. The crayfish are slightly smaller, and they use their claws to hunt smaller prey, like worms they dig out of the lake bed, or insects they pluck off down from near the surface of the water. Crayfish are also important consumers of aquatic plants, keeping them at bay so they don't crowd the water surface and cause eutrophication. Altogether, these are the largest living animals with the caracoid facies body form. Compared to the smaller shrimp, prawns, and krill, the Astacidea are big and beefy, with robust legs, a thick exoskeleton, and large, imposing claws on the first set of legs. To illustrate this size difference, consider the euphausiids, the krill. The largest specimens of the largest krill species can grow up to 15 or 16 centimeters long. The largest prawn species is the giant tiger prawn Peneus monodon, which can grow to 28, 29, or even 30 centimeters long. In the imperial system, that's about 6 inches for the biggest krill and 12 inches for the biggest prawns. Meanwhile, the typical Astacid lobster can grow anywhere from 20 to 50 centimeters. They can potentially grow larger than that, as they're really long-lived, with lifespans measured in multiple decades, and they can keep growing and molting more or less the whole time. As a result, some of the largest individual lobster specimens are over a meter long, and 70, 80, 90, even 100 years old or more. Due to their long lifespan, there are rumors that lobsters might be immortal. They're naturally long-lived. They grow more or less continually throughout their life, and unlike so many other crustaceans, even some cephalopods and fish, they don't reproduce once and then die. Recent molecular analysis has revealed that lobsters have telomerase enzymes that are expressed throughout adulthood, repairing their DNA after every cellular division and during every molt. This basically means that their cells stay healthier, so their tissues and their organs stay healthier. They're not really suffering from any significant genetic or cellular degradation as they age. They're not suffering from senescence, so their metabolism and their cellular activity should all be theoretically sustained at healthy levels indefinitely. They should all be composed of genetically stable lineages of cells. Put this all together, and it's understandable how someone might think that lobsters could theoretically live forever, so long as they aren't killed by a disease or a fungal infection, or chewed up and ripped apart by a hungry predator. Unfortunately for the lobsters, this theoretical possibility is not so. they are not immortal. Although they are quite long-lived and genetically healthy due to all of the telomerase activity throughout adulthood, although they don't seem to slow down or weaken with age, and in fact seem to get more virile as they get older, their death is ultimately guaranteed by the limitations and structural flaws of their complex exoskeleton. In the first year of their life, they can go through dozens of molting events. As they age, the frequency of molting events slows down, so maybe a few times a year, and then once a year, and then once every 2 or 3 years. After many decades, molting events may occur only once every 4 or 5 years. This is a pretty long time interval, and the exoskeleton may accumulate a lot of damage. When it's younger and it's shedding its exoskeleton when it's molting multiple times a year, there's not enough time for the exoskeleton to build up a lot of damage. By the time there's enough, scratches and nicks and dents worn up in it, it's time to shed it and get a new one, so they never build up that much. But when they're older, and these molting events occur every half decade or so, they do accumulate a lot of damage, a lot of cracks or puncture wounds. Sections might be fractured or flaked off. Bacteria can seep in through these gaps in the exoskeleton, and they can infect, inflame, and damage the softer flesh inside. Scar tissue forms, and this can often fuse the flesh to the inner surface of the exoskeleton. When the next molting event comes around, the exoskeleton won't detach completely. If it's ripped off, soft flesh can be ripped up too. Scar tissue is basically torn out, and this can lead to massive, gaping, and often fatal wounds. If it's not ripped off, and the lobster just struggles in the middle of a molt, it can get stuck. It can run out of energy, and it can starve and die. In extreme cases, the exoskeleton itself can start to rot, and the lobster, trapped inside of what is essentially a plague coffin, dies of full-body sepsis. Crayfish, on the other hand, being smaller and generally closer to the surface in their freshwater, riparian, and littoral habitats, are prey for a much wider range of animals, including mink, otters, muskrats, all manner of seabirds, as well as larger fish like pike, snakeheads, perch, and eels, as well as larger crustaceans, including fellow astacids. Add to this their vulnerability to water pollution, their sensitivity to salinity and water oxygen levels, and their susceptibility to a highly lethal water mold infection called crayfish plague, and the fact that larger crayfish have all the same issues with molting and exoskeletal breakdown that lobsters have to deal with, and you can understand why many crayfish species typically have shorter lifespans than your average lobster. But make no mistake, the crayfish are still very long-lived for arthropods. For example, the Australian Murray crayfish, Eustasis armatus, reaches sexual maturity after a decade. And then you have absolutely wicked anomalies like the Tasmanian giant freshwater crayfish, Astacopsis goldii, which reach sexual maturity after a decade and a half, and which can live up to 6 decades and grow to be almost a meter or over 30 inches long. This anomalous crayfish, the Tasmanian giant freshwater crayfish Astacopsis goldii, is larger and older than a pretty decent number of nephropid lobsters. At the other end of the size spectrum, you have species like the swamp crayfish Tenwebrinchiurus glypticus, which is a mere 25 millimeters, or a single inch long, with a similarly reduced lifespan and life expectancy. This diminutive species is much closer to the base of the food web and serves in an ecological role roughly analogous to a freshwater version of krill. One of the most characteristic, distinctive, and beautiful aspects of the astacids is the coloration on their exoskeletons. Not only is there an incredible diversity of colors and patterns across all the astacids, there's also a number of mutant phenotypes that are really exotic, or strange, or just really badass. Most species of crayfish and lobster are earth-toned to blend into the seafloor or the bed of a river or a lake. These can be various subdued shades and mixtures of tan, beige, brown, black, gray, rust, green, and even blue. Many species have a scattering of spots along their claws and back, highlighted edges, or beautiful marbled patterns. Sometimes the claws or the forearms or the legs are a different color from the rest of the body. That enormous Tasmanian giant freshwater crayfish is a muddy brown color, but its primary forearms and its large claws are a pale bone white. There's also particular mutations that we've observed in several types of astacids, and these create really amazing colors. About 1 in every 10 million to 1 in every 30 million lobsters will have an individual who is a bright red, a startling orange, or an almost neon yellow. Bright blue colors are an order of magnitude more common, whereas albino lobsters and glassy opal-like so-called cotton candy lobsters are an order of magnitude less common. Sometimes there's mutations in the gonads or reproductive systems, leading to hermaphrodism and a split coloration. In this condition, the lobster may have a perfectly straight dividing line running from the tip of their head to the tip of their tail, the right and left halves of the body being different sexes, with strikingly different colors. And it really is surreal how these strikingly different colors meet together in a sharp, clean line running perfectly down the middle of the lobster. It's one of the most unusual and wildly surreal things to see on an animal that's otherwise quite rugged, bumpy, and spiky. Well, there you have it, folks. That was a fun exploration of the biology, ecology, and morphology of the lobsters, the crayfish, and their primitive relatives. This was a super interesting episode, and if you enjoyed it, hit the like button, or give it a 5-star review. In the next episode, we'll be looking at the final branch of Reptantia, the crawling decapods, by exploring the biology, ecology, and morphology of the Brachyura, the true crabs, and all of their more primitive cousins. If that sounds cool to you, then subscribe to the channel to get that new biological content right when it drops. Your likes and your comments and your subscriptions really help me in the algorithms, and it really helps get the show out there to more people. Now, if you want to support the show financially, check out the official store, or become a Patreon supporter. The tiers on Patreon are as cheap as $1, $2, or $3 a month. And if you think that's worth it for one show a month, then by all means, sign up and become a patron. However you support the show, it means the world to me. I can't do this without you guys. And as always, thank you so much for listening.